In this lesson, we will examine membrane transport. We will focus on how the structure of the plasma membrane determines how substances move into and out of the cell. Understanding membrane transport is essential for understanding homeostasis, cellular communication, and tissue function. The plasma membrane is composed of roughly equal amounts of lipids and proteins with smaller amounts of carbohydrates. The lipids provide the structural framework, while the proteins perform many of the membrane's specific functions. Its basic structure is a phospholipid bilayer. Each phospholipid contains a hydrophilic, or water-attracting, head and hydrophobic fatty acid tails. The heads face the watery environments inside and outside the cell, while the tails form the membrane's interior, creating an oily barrier. Embedded within this bilayer are membrane proteins. Some proteins extend completely through the membrane. These are called integral proteins. Others are attached to only one surface, known as peripheral proteins. These proteins function as channels, carriers, receptors, enzymes, or structural anchors. Their placement within the membrane allows them to control what enters and leaves the cell, and how the cell communicates with its environment. The hydrophobic interior allows lipid-soluble molecules, such as oxygen and carbon dioxide, to pass through easily. However, water-soluble substances, including sugars, amino acids, and ions, cannot cross directly and often require these embedded proteins. Cholesterol molecules within the membrane add stability and help regulate fluidity. Because lipids and many proteins can move laterally within the layer, the membrane is described as a fluid mosaic, stable in structure, yet dynamic in function. The structure of the plasma membrane gives it a property known as selective permeability. Because the interior of the phospholipid bilayer is hydrophobic, small nonpolar molecules can pass through easily. Substances such as oxygen and carbon dioxide dissolve in the lipid portion and diffuse across the membrane. In contrast, large polar molecules and charged ions cannot move freely through this oily interior. These substances require assistance from membrane proteins. Selective permeability allows the cell to regulate what enters and what leaves, maintaining internal stability. Passive transport is the movement of substances across the plasma membrane without the use of cellular energy. No ATP is required. Instead, movement occurs because molecules possess kinetic energy. They are in constant motion and naturally spread out from areas where they are more concentrated to areas where they are less concentrated. This movement down a concentration gradient continues until equilibrium is reached. Passive transport includes simple diffusion, facilitated diffusion, and osmosis. Each process depends on the structure of the membrane, but does not require energy input from the cell. Simple diffusion is the movement of small, nonpolar molecules directly through the phospholipid bilayer. Because the interior of the membrane is hydrophobic, substances that are lipid-soluble can dissolve in this region and pass from areas of higher concentration to areas of lower concentration. This process does not require ATP. Movement occurs naturally as molecules spread out down their concentration gradient. Oxygen, carbon dioxide, and certain steroid hormones are examples of substances that cross the membrane by simple diffusion. The ability of these molecules to pass directly through the lipid portion of the membrane is a direct result of membrane structure. Facilitated diffusion allows water-soluble molecules, like glucose or ions, to cross the membrane with the help of specific proteins. Channel proteins form pathways for ions or water, while carrier proteins transport larger molecules, such as glucose. For instance, glucose binds to a carrier protein, which changes shape to move it across. Although this process is highly specific, each carrier only works for certain solutes, and limited by the number of proteins, it still moves substances down their concentration gradient without ATP. Osmosis is the movement of water across a selectively permeable membrane toward an area with a higher concentration of solutes that cannot cross the membrane. In other words, water moves toward where impermeable solutes are trapped. If there's a difference in solute concentration on either side, water will naturally move to the side with more of those trapped solutes. In short, water follows solutes. While there are various detailed explanations of osmosis, for now, just remember that water will always move toward the compartment with the higher concentration of impermeable solutes. Osmosis ensures that water balances throughout the body. In both intracellular and extracellular fluids, water concentration is essentially equal, meaning the osmotic pressure inside and outside cells matches. A solution with the same osmotic pressure as body fluids is called isotonic. In an isotonic solution, cells remain stable and do not change size. Active transport moves substances across the membrane against their concentration gradient. Because this movement goes from lower concentration to higher concentration, it requires the cell to expend energy, typically in the form of ATP. Unlike channel proteins, which allow substances to diffuse down their gradients, active transport relies on carrier proteins. These proteins bind to specific solutes and use energy to change shape, transporting them across the membrane. Active transport enables a cell to maintain internal concentrations that differ from those in its surrounding environment. A key example of active transport is the sodium-potassium pump. Compared with its surroundings, an animal cell maintains a much higher concentration of potassium ions inside the cell and a much lower concentration of sodium ions. The plasma membrane preserves these gradients by pumping sodium out of the cell and potassium into the cell. This process requires ATP and is essential for maintaining proper cellular function. Bulk transport moves large molecules, such as proteins and polysaccharides, as well as larger particles, across the plasma membrane. Because these substances are too large to pass through channels or carrier proteins, they are enclosed within membrane-bound vesicles for transport. Like active transport, bulk transport requires energy from the cell. Endocytosis allows the cell to bring in biological molecules or particles by forming vesicles from the plasma membrane. Though different proteins are involved, the process looks like the reverse of exocytosis. A portion of the plasma membrane sinks inward, forming a pocket. As the pocket deepens, it pinches off, creating a vesicle inside the cell that contains material from outside. Exocytosis is the process by which materials are exported from the cell. A transport vesicle, typically formed from the Golgi apparatus, moves along microtubules of the cytoskeleton toward the plasma membrane. When the vesicle reaches the membrane, specific proteins facilitate the fusion of the vesicle membrane with the plasma membrane. As the two lipid bilayers merge, the contents of the vesicle are released to the outside of the cell. The vesicle membrane then becomes incorporated into the plasma membrane. The plasma membrane structure determines how substances move. The lipid bilayer forms a selective barrier. Embedded proteins provide controlled pathways. Concentration gradients determine direction. Together, these mechanisms allow cells to maintain internal balance. Thank you for watching. If you found this helpful, please like and subscribe.